To introduce to Dr. Yan He from China Agricultural University. I met Yan, I think it was 2007, the first time, something like some times ago. We did our postdoc in the same lab, in the lab of Dr. Wojtek Pawlowski in Cornell University. So Jan told me that he did his PhD in Florida, and after went to postdoc from 2000, right?
Yeah, 2010, 2013, yeah.
13. Yeah. And from then he got a position as a leader in the China Agricultural University. That is one of the biggest university for agriculture in China, in Beijing. Jan has published several very interesting paper related to maize meiosis. He's one of the experts. In the last 10 years, probably most of all meiotic genes from maize come from his lab. But he diversified also his research and have a wider interest in agronomy in general, principally on maize, but not only. So it's with great pleasure that I welcome Jan to give his presentation. Thanks.
Oh, all right. So can I start?
Sure.
All right. So thank you. So yeah, thank you very much for the invitation for Arnold. It's my great pleasure to have this chance to give a presentation. So I just slightly changed the title for my today's talk, because for the maize meiosis, it's really old story in my life. So actually my life now focusing on many other trade in maize. So I would take this chance to give a several story about the new work in my life. So the title for my today's talk is Genetic Control on the Natural Variation in Pollen Number and Fertilities in Maize. So as introduction from Arnaud, in the last 10 years, my life mainly focusing on the male meiosis. We are interested in the double-strand break formation and DSB repair and eventually crossover. So we are characterizing several genes during this pathway, either from DSB formation and repair. So several work have been published. However, several work still in the progress. And also the second second aspect of research in my lab is we are also work several projects in the epigenetic and IP transcriptomy. So we have working on the one of the very important regulator in DNA methylation called the DDM1 for several years. So all of those genes was with a complex with DDM1. Now we are starting our try to characterize several genes interacting with DDM1. But as I said, some of them was published, some are still in the middle. So as I said, I would like to introduce several new work in my lab. So today I bring three small stories. The first, I'm going to introduce the Maize ASY1 can interact with PRD3, and this module is really crucial for meiotic double-strand break. And the second, I will going to introduce the one of the interesting gene called RPIN1, which is the first gene to regulate the natural variation in pollen number. And also I will introduce how to use this gene to boost the hybrid seed productions. And lastly, I'm going to introduce another two genes called gene 1 and MSH7, which control the natural variation in pollen fertilities. So let's go to the first one, ASY1 stories. So if you guys are familiar with the maize meiosis, we know that ASY1 is a protein called axial proteins. And also PRD3 is crucial for double-strand break. It's a double-strand break forming proteins. However, you know, if you look at the different species, including the yeast and the moth, we know that the double-strand break was formed along the axial. But however, if we do the bioinformatics study, we know that those DSP side is in the chromatin loop, not in the chromatin axial. So this trouble, you guys imagine that with the DSP side have to be transferred from chromatin loop into the chromatin axial. So what's the mechanism behind it? What kind of protein? What kind of complex can work to recruit or transfer the DSP side from chromatin loop into the chromatin axial is a remaining mystery in maize. So this is our question. So in the yeast, there are several proteins involved in this step for recruiting, including the MER2, REC114, and the MAY4, several others. And also in the mouse, there are also several proteins involved in this handling. So if you look at the homolog of those genes in maize, we know that ASY1 is the HOP1 homolog in yeast and HOMA in mouse. Also, we know that ASY1 is the axial protein. And also we know that PRD3 is the MER2 homolog in yeast and IH1 in mouse. So all of those homolog was conserved in maize, but you know, different species utilize different mechanism to conduct this tantling. So we are going to ask if those homolog in maize was have a conserved function for this tantling process. So the first we identified the ASY1 mutants in maize. So we obtained several different alleles for these genes. And for the knockout mutant for ASY1, they show us both male and female sterility. But that's really normal for the mitotic gene in maize. Also, if we go further to see the chromatin behaviors, we see that in ASY1, there are a lot of univalent can be formed in the mutants. That's very interesting because suggesting us those univalent may be from the defective DSP formation. So then we go to further to observe if the DSP was abolished in the mutant or not. So we're using the two markers, one called gamma2AS and the second called RED51. Both of the two markers is the typical marker for testing the formation of double-strand break in maize. So when we do the immunolocalization, we can see that compared to wild type, both of gamma2S and RED51 signals was significantly less than the wild type. So we make conclusion that ASY1 is required for not essential, but very important for the mitotic DSB formation. And also we raise a specific antibody against the ASY1, and we can see this ASY1 can load normally in wild type, but it's disappear in our AS1 mutant. And also the central element, the JYP1, was abolished for their loading on the chromatins. So right now we can make a conclusion that ASY1 is really important for the double-strand break formations. Then we are going to make a test to make an attempt to see why ASY1 is required for double-strand break. Also, so we are trying to screen the interacting protein with ASY1 because the question is we think ASY1 can be interact with one of the DSB forming proteins. So we test this. So we use the ASY1 as the bait in the EST2 hybrid system, and using all the other several DSB forming protein as the bait. You can see from this result that the ASY1 can only interact with the PRD3. Also, so this is very interesting, as I said in the introductions. Okay, so you can imagine that ASY1 as an axial protein can interact with DSB forming proteins. So we confirm this interaction in both ES2 hybrid and the LUX system. Then, because we already found that ASY1 may interact with PRD3, then we go further to characterize PRD3. So we also obtain several mutant alleles for these genes, and all of those alleles also show us the completely male and female sterility. That's really normal. Then we go further to see there's chromatin behavior. We see in the PRD3 mutants, all the bivalent was gone, was disappeared, and with the 20 univalence, that's the typical for the DSB defect mutant. Then we go see the gamma2S and the REB15 marker is so as complete no signal. For those results, we can make a conclusion that also we do the double mutants. We use the PRD3 mutant, try to rescue the RED51, the chromatin fragmentation phenotype in the RED51 mutant. So based on the chromatin behavior, based on the immuno, and based on the double mutant result, we can make conclusion that PRD3 is essential for the DSB formation in maize. Then we already know the PRD3 is both PRD3 and the ASY1 is important for the DSB formation. So we try to ask question: what are their localization in the chromatin? So we reach we have the AS1 antibody. We also have the PRD3 antibody. So we try to do the co-immunolocalization, find the results so that we can see PRD3 can co-localize with ASY1 only on the chromosome axis. And meanwhile, the loading of ASY1 and the PR3 on the chromatin is independent to each other, which means ASY1 signal still on the chromatin on the PR3 mutant, and on the other side, PR3 signal can be still maintained in the ASY1 chromatin, which means the PR3 and the ASY1 they are localized on the chromosome in the independent manners. So we try to help these models. Okay, so we do a lot of the East to hybrid screening for their partners. So eventually we have this model that the ASY1 and the PRD3 can work as a key module, which link DSB site to chromosome X during the DSB formation in maize. This mechanism is quite similar to the same models described in yeast and mouse, indicating that this tunneling process may be evolutionarily conserved in the very different types of species. So we have this work published just last month or 2 months ago in the New Phytology. Okay, so this is the first story I would like to bring today. Then I go to my second story. So this I don't know if everybody in this familiar with maize or not. If not, at least you can see the maize plant, right? This is the tassel. This is the male part for the maize. Okay, so they have a long branch, and each branch have a lot of spike, and each spike have their 2 florets, and in each floret there are 6 answers. So I would ask a question that, the maize is very, very popular, right? We have maize no matter in each country, in a lot worldwide, we have maize, and those maize have a lot of diversity. Okay, so they can grow in different place, right? So the question is, do you know if the pollen produced by a plant, how many pollen can produce per plant? That's a really tough question, because we know that each maize plant can generate thousands and thousands pollen grain. Okay, so my question I will ask is there natural variation in the pollen number in different plant species? This is a mystery. Nobody knows that. So for addressing this question, we have to have a way to count the number of pollen with a high throughput. Okay, so one of my students is quite smart, so we figure out a way to quantify the number of pollen per plant with only 10 minutes. Okay, for each plant. So we have this method published last year. Sorry. Then we go further to use this method to count one of our device population, genome-wide association population. This population consists of more than 500 different inbred land, which collected from different places around China and across the world. Okay, so we have this association population. We count the pollen number for each land. Then we make this device analysis. In the chromosome 9, we found one of the most significant signals. These signals locate on the gene called RPN1. We named this gene as RPN1, regulator of pollen number 1. So we know this is the most significant association with pollen number. Then we do the more detailed sequencing, try to address what's the causative variant to affect this gene. So after that, we found an indel. This indel quite large, is a couple hundred base pair located on the promoter of RPN1 genes. So if this large indel variation can change the transcription, can change the RPN1 transcription. Okay, so and then we based on all of the variants, including SMP, indel, all the other variants, we have the haplotype analysis. So we found that haplotype 1 always have the higher number of pollen number than the hap2. Okay. Then we go further to characterize this gene. This RP1 encodes a membrane trafficking protein. We double tracked it using our fluorescence test, and then we do this gene was highly expressed in the male in the tarsals. Also, we obtained several mutant alleles for this gene, and after counting pollen number, we found that in the mutants, the number of the pollen number was significantly increased compared to wild type. Then we concluded that this gene can negatively regulate pollen number. And also we do the in situ hybridization. We found this gene was mainly expressed in the torpedo cell, okay, during the anther development. And also we use the other method to confirm the number of the pollen. So we found that along with the change of the pollen, the torpedo cell was also altered. So it seems that this gene can regulate the balance between the torpedo and the mitotic cell and the pollen cell. Then we try to figure out what's the molecular mechanism behind the regulation of RP1 with pollen number. Then we do the large-scale East2 hybrid screening. We found an interacting protein called MSP1. This is a really famous gene. It's a gene controlled in different plant species, which have been known to regulate the number of the torpedo cell and the pollen numbers. This is already been noted to affect this process. Then we confirm this interaction in the East2 hybrid and the log system, and also we use the in situ hybridization to make sure the MSP1 does also express in the torpedo cell. And the mechanism we are trying to figure out is because RP1 encodes a membrane trafficking protein, which can help or facilitate MSP1 to locate and localization to the plant members. So this the mechanism for the how RP1 regulate the number of pollen. And also we try to use this gene in the practical breeding. You know, the maize is the hybrids, right? So for the commercial company, they have to make hybrid seed production. Then they sell those seeds to the farmers. So during the generation or production of hybrid seeds, they have to use the female and the female as separate. So normally in this process, one male with the full rows and the female was taken for the routine. You know, so if you can imagine that we can increase the pollen number for the male part, right, then we can increase the ratio of between the female and the male for like 1 to 6 and 1 to 8 rows. So we took these ideas, we make this really strict field isolation test. We found that, you know, using the RP1 mutant, we can guarantee the production of seed, you know, in the field. So all this story have been finished. It's going to be published. Actually, it's already been accepted. Okay, so you guys can have this manuscript maybe next month or something. Okay, all right. So then I move to my last story. I'm going to introduce today is also one of my most important funding in my life during the last several years. So in the process to count pollen numbers introduced in my last project, so one of my students occasionally observed that not all the pollen in the natural inbred land was fertile. The story, in fact, most of the inbred land for maize are not 100% of fertile. They are naturally sterile for some certain degrees. So then we count this fertility. I have to emphasize that this is in the normal condition. Non-mutants is just the natural inbred land. Okay, so we examine the fertility for our association population. We found that this fertility in different land is quite distributed as a normal distribute. Then we go further to make the GWAS analysis to see what kind of gene regulate the natural fertility. So we found the two genes, MS11 and G1, as the most association genes with the pollen fertility. I was really surprised by these two genes because I'm working on the DSB repair. Both of the genes was known to involve in the DS repair process. Okay, so this also reminds us that a lot of natural variant in the DSB repair-related pathway can be associated with a lot of human illness, right? So a lot of cancer, whatever. But in plant, what kind of natural variation, what kind of natural variant in the DSB related gene can contribute to each of the plant trait is unknown. So we are really surprised to see this result. Then we go to characterize those two genes. For the gene one, after we have the mutants, this gene have two copies, 1A and 1B. So we have the mutant for those two genes. And then we can see that in the mutants, the mitotic division in the sperm development was aborted. A lot of DSB was formed in the sperm, in the mitotic divisions. So this gene, so we conclude that gene one can evolve in the sperm development in the pollens. Then we have the MS7 mutants. So this gene is different from gene one. So after we see the cytology result, we make a conclusion that MSCT control can regulate the normal process of meiosis. So it seems that we have two genes contribute to the pollen fertility in the natural conditions, and both genes are involved in the different process of male development in maize. To our surprise, is we're thinking that as I introduce, each maize plant can produce thousands and thousands pollen. So the question we ask is, if we decrease the number of the pollen generation, is this can trigger the relocalization of the nutrition from male to the female? Okay, so in the long time breeding history, the farmer can always observe that the high yield hybrid can always associate with small tassels, which give us a hint that this relocalization or competition of nutrition between female and male exists. Then we see our mutant. We found that because MH7 mutant have a decreased pollen fertility, it's not completely fertile, it's just decreased. So we want to investigate if this decrease in the pollen can increase the female production. It does. We do several years field yield tests. We found that in the context of MS7 mutants, either inbred land or the hybrid background, this mutant can significantly increase yield by 5 to 8. And also very interesting that this increase largely from protein increase. So we have this story going to be published, maybe also next month. So hopefully you guys can like it. So I probably it's a half hour is great. So I finish my talk today. So I would like to give my appreciation for all of my students. As you guys know that in China, we always have a huge lab. So now ask me how many guys in my lab. I told them right now I have more than 5 postdocs and more than 20 graduate students. So we do have a huge lab. So we do have a lot of we have several grants supported from National Science Foundation and the National Key Program, and also I have a lot of collaboration with some company, including Xinzheng, whatever the others. So I will give my highest appreciation for all the supporters. Okay, I stop there. I would like to ask any question if you guys have. Thank you.
Do we have any question? So I will start with a technical one. To check, well, you have a lot of experiments that check for protein-protein interaction. Mm-hmm. And in some of your last publication, you mentioned that you use luciferase assay instead of the BYFC. Do you see any reason to do that?
Mm-hmm. We do have several systems to test the interaction between the proteins. So you know so the log system work more is more simple to work on it. Yeah.
You haven't tried the BEFC experiment?
Yeah, we do try. Yeah, we do try. Yeah.
Just a choice of experiment, right?
Yeah. But you know, if we want to make 100% sure about the interaction, each log or the defective is not. I'm trying to say we have to test several different systems to get a solid data.
Because basically most of the experiments done in Arabidopsis are confirmed with what you observe in Maize, but there is some discrepancy, and I wonder if it's a question of experimental tools that are different.
Not different. It's a worthwhile in Maize. Yes.
So there's a question from Alejandra Covarrubias in YouTube. Which are the identity of gene 1 and MSH7?
Can you repeat? Sorry.
Which are the identities of gene 1 and MSH7?
What are the? So I can't hear you very well. What other what?
Which are the identity of gene 1 and MSH7? So, well, to answer Alejandra, there are known meiotic genes as well.
Let me go to For the gene 1, it's a repair related gene. According to the report from Arabidopsis and Rice, is involved in the homologous recombination pathway, but we just see that this is a repair only involved in the mitotic division, in the spring development, not in the mitotic, not in the meiosis. However, for MS7, is a protein involved in the meiosis. Yeah.
And you have another question from Pepe Reyes. Thank you for your presentation. Have you found protein interacting with APN1? And if so, are there any that also affect pollen production?
Okay, I go to the, yeah, we do have the have this interaction between RPN1 with MSP1. So MSP1 is a gene have been characterized in maize several years ago. The phenotype do show us the increase in the pollen number with the decrease in the torpedo. Yeah, we do have this phenotype for the MSP1. Yes.
Any more questions from the YouTube? So thank you very much, Jan, for your presentation.
No problem.
I hope we can see each other again soon. Bye.
Yeah. Thank you so much, everybody.
Bye-bye.
Bye-bye.